Okay, hello everybody. I hope you can all hear me. So today I'm happy to share that today one of the Hebrew University's close friends from Italy, Viviana Kassam, will introduce, you probably have understood it, Dr. Naomi Habib, who is the Goren-Kasam Lecturer in Brain Science. Viviana is Honorary Fellow of the University and the President of Brain Circle Italy and Lugano. Viviana, please.
Well, first of all, let me tell you how happy and honored I am to present Naomi. She's a great woman, or a great scientist, but also a great woman. She has 3 kids, and I am very much pro women in science, but it's also good to have a woman in science who has a full life and can be a mother and do outstanding studies. And I am very proud because Naomi, as Mara said, is holding the chair I created, and the chair was created for her. And so to see her success and her great results in her studies and her research makes me full of joy and pride, and not only me, but my whole family. And I'd like to take a few minutes to tell you how this idea of a chair for her was started. The chair was created for her. I wanted to do something for the Hebrew University. I did not know what to do. I had many ideas. And then a great friend of mine, Mona Sorek, called me and she said, listen, we have this fantastic scientist. We want to get her at Hebrew University. Weizmann wants her. I don't remember if Harvard or another great American university, was it Harvard? Yes, wants her. No? And she said, we really want to have her and we need to make a chair for her. Are you willing to do it? And I said, great, if she's a woman, you know, because I promote women. Brain Circle Italy was started with me and Rita Levi-Montalcini, the Nobel Prize. And her idea was that we have to disseminate science and also to promote getting women more interested in science, which might not be the case in Israel. I know you have much more scientists than we do in Italy, but in Italy, it's really needed. And so, you know, I didn't hesitate a minute. I met briefly by email and then Skype with Naomi. I was really impressed with her. And I want to tell you, and this is very important in this moment where there is an economic crisis and we're all losing money and very worried, and to make an investment in the Hebrew University and in science, and I mean in any other institution, but my feelings are for the Hebrew University, is the best investment you can make. Your money is safe. Your money is growing. Your money is producing. And this is wonderful. And also it makes you feel very good. So it is my encouragement to everybody. Donating makes people happy, makes you feel worthy. And I want to say one more little thing, that this was a bigger donation. I made also a small donation to the Hebrew University for a student from India. There is a program that teaches students how to make policies for the third world, health policies. So they go back and in their country they implement what they learn for public policy, which means helping not one person, but the whole country. And it was a woman also, of course, that I helped, an Indian woman. And she was promoting in India, was studying because she suffers from diabetes and she wanted to go back to India and make a big program for diabetes. As you know, COVID is very, very dangerous for people suffering from diabetes. And she also made a program to help these people. And it's another thing that made me feel very proud and made it so much worth the donation. And so I just wanted to say these 2 things. And Naomi made a great study and a great research, which is going to be, has already been published all over the world. Bravo, Naomi, keep on like this. And thank you to everybody. I was asked to say that the webinar will last half an hour and then there will be time for questions. Thank you.
Thank you. Thank you, Viviana, for this really great introduction and your ongoing support for science and women in science, and for me and my lab in particular. So I'm greatly honored. Wanted to say that we're now at a special time where scientific contribution is especially meaningful. And I see around the world the involvement of scientists who really have been putting everything aside and gearing up to handle this very unique situation of the, oh, we have a little runaway here. That's working from home during these times. So I get actually every talk I give, there's a little visitor who comes in. So I wanted to say it's very exciting, and specifically actually Viviana has shared with me that she, as usual, she's very, very busy, and she has built an amazing website that actually communicates science to the people. It's of course in Italian, it's designed for Italy. But I've looked at it, and this is actually one of the things that are greatly needed, the way to communicate science to the public also. And so thank you for the Friends of the Hebrew University for giving me the opportunity to tell the story of our science and what we are trying to do in the efforts to get out of this crisis of the COVID-19 pandemic. And I see that it was also a great opportunity for me to invite family around the world to also hear what I'm doing usually. And I see there a lot of them have joined us today, cousins and my parents and my family from around the world. So I'm especially excited today. So let me just start by sharing this presentation. I hope you can see it. And I'll tell you today about how we've been trying to address the COVID-19 crisis. You just have to move this aside. Okay, so I hope you can see this. If there's any technical issues, please let me know. Okay, so I'll tell you a little bit about myself. In my daily life, when I'm part of the LSEC Center for Brain Sciences at the Hebrew University. This is where I built my lab 2 years ago with the generous help of Viviana. And our focus is really on understanding the brain. We're studying aging and Alzheimer's disease, and we're trying to address critical questions in the understanding of aging and Alzheimer's. So this seems completely unrelated to the COVID-19 pandemic, but actually, as part of our work in order to understand the brain, we've been developing new technologies, genomic technologies, to measure RNA molecules in single cells. And I'll get back to this later and explain more, how is this actually related to COVID-19 testing? And really, at the beginning of February, we've realized that the world has changed. And we're now in the midst of the COVID-19 pandemic. In the midst of March, we've also been in Israel. We had to all be home for a very long time. And now it's finally being kind of a little bit more open, but looking around the world, we see the pandemic is still very strong. It's been affecting our lives. I don't need to tell that to all of you because you've been living that along with me. We see how it affected millions of people around the globe. We've seen too many lives have been taken. And there's also a huge toll, both emotional toll on people and an economic toll for many people who have lost their jobs and their money. So there's really a huge need to find a solution for this pandemic. And to me, the way out of this pandemic is testing. Testing, and more testing, really massive testing. And why is this so important? it's important in order for us to go back to our routines, to go back to our work, to go back for the children to go back to school, and to prevent second and maybe even third outbreaks of this virus. And why is that? Is because if we're conducting massive testing, we can find and really identify early any signs of outbreaks of this disease, and then contain these outbreaks, track infection chains in order to prevent the infections global-wide. That would mean that we could all kind of return to our daily lives while individuals who have been affected will be contained at home for the time needed. So this is actually a model that's been used, for example, successfully in South Korea. And you can see here a picture of like a phone booth test clinic in South Korea where people can go through and get tested. And if you just like even a quick search in the internet, mass testing has produced so many titles from many, many articles all saying the same, that in order to get out of this pandemic, in order to finish the pandemic, in order to get back to our normal routines, we have to do mass testing. So why is this not being done today? So we could really identify two major roadblocks. And the first is that we are missing the materials, the reagents, in order to do the standard diagnostic tests. We don't have enough of them in order to do the millions of tests needed worldwide. And the second is that the current tests that are being done are accurate, but they're not efficient. They're not scalable to the scale we need. And these are the two goals that we have set up for ourselves. When I say we, let me introduce our team, because it's been really an amazing adventure. Oops, I ruined the surprise. Sorry. So the team is a very tight collaboration with Professor Nir Friedman from the Life Science Department at the Hebrew University. Just a small disclosure, so Nir was actually my PhD advisor when we were both at the Computer Science Department, and it's quite a lot of years ago now. And we've all each gone to a very different way, but we do have a similar core of thinking of technologies and genomic technologies. And that's what actually brought us together in order to develop these new therapeutic testing. And we have an amazing team of over 15 scientists really working day and night on this project. You can see here, I've been highlighting people from Nir's lab, Ayelet, Alon, Isra, Ronen, Matan, Dafi, and in my lab, Masha, Miriam Adam, Danny Kitzberg, Anna, Christina Schmidberg, and Anael Kain, who've been, it's been such an inspiring, so inspiring to see them putting all this amazing effort together. And there is actually, it was not a trivial thing to ask them to do, because we all have families, and the families have been in their own kind of crisis, confined at home, homeschooling, and all these challenges. So I want to get this opportunity to actually thank the most important supporters of this effort, which have been our families. So I'm actually only exposing my family here. It's a great opportunity support group, so to my husband, Ariel, to Daniela, Amos, and Galia, who've been amazing. And if you are watching this webinar, kids, it's time to go to sleep now. But it's been really amazing, and it's nice to see that even small children can understand the importance during this time of getting recruited to fight the pandemic. So let's get to what we actually did. So I'll give you a very quick overview of the current diagnostic tests and their limitations. I'll describe the collaborative efforts at the Hebrew University with Hadassah Hospital, which have been actually critical for this project. And then I'll describe two steps: our published improved homemade COVID-19 diagnostic test, and the next generation scaling up COVID-19 diagnostic test using next generation sequencing, which we think will be really useful for mass testing. And if you're going to leave this webinar early, I just want to kind of start actually with my take-home message, which is scientists, and this is true actually worldwide, we've been talking with colleagues around the world, are really developing innovative technologies and solutions that are paving the way to conduct the mass testing that was greatly needed and missing around the world. So this means that that we're doing our part, the scientists, we're going to deliver the technology to enable mass testing, and we really need the policymakers to join us in these efforts to actually implement them and make sure we are doing these massive testing that are needed. So how are the current diagnostic tests done? So when I'm talking about testing, I mean testing that are done in the labs. There are a lot of innovative ideas that might bring up other solutions. For example, tests that somebody can do, like a 5-minute test you can do when you're about to board an airplane. And these are all really needed, but these won't probably be used for the massive testing we're talking about. There's also a very big discussion about antibody testing that I think will be complementary efforts to what we're doing and are also very, very critical, but currently they lack the precision that we are needed, and we hope for also scientific breakthroughs in that field as well. So how are these testing being done? Just do this. Okay. So we get the samples from the individuals after they were swabbed in their mouth in small tubes. And these tubes contain the viruses, the live viruses. And within the viruses, there are the genetic material of the virus, which comes as a molecule that's called RNA. So the first step is to add a buffer that completely destroys all the proteins and all the viruses. And so releases these RNA molecules. So we have these precious RNA molecules we're looking for, but they are surrounded with a lot of mess around them. So the first step is actually cleaning up the RNA molecules from all the other materials that comes within this test. And then we take these the RNA molecules and do a process called real-time PCR or qPCR to actually measure the RNA, and we're asking a very simple question. Can we find a viral RNA or no? So we can give the person an answer if he's sick or healthy. So this is the general idea, and as you can see here, we're doing this test, and this is quite common in a plate that has 96 small wells, which means we're actually processing that we have the potential of testing 96 people at a time. So this sounds pretty good, but the problem starts mainly start at this level when we try to extract the RNA molecules from all the rest surrounding them, cleaning up the RNA and extracting it. So this RNA cleanup has two problems. It's very inefficient largely, and there's a huge shortage in reagents. So actually this project started when I got emails from my colleagues in Boston who were working in labs in Boston who shared with me that hospitals around Boston were asking them for materials to extract RNA. And that got me really scared because, you know, if hospitals in Boston, you know, in my view, if hospitals in Boston don't have these reagents, of course in Israel we won't have these reagents because we're not producing them in-house. They're all produced overseas. So we immediately tried to think of alternative solutions for these two great issues. One is making this RNA process faster and doing it with an alternative way. But let me first describe the setup where we are at part in the Hebrew University. So the Hebrew University has actually done something amazing, and this is largely an effort led by Professor Yuval Dor, and where he started a tight collaboration with Professor Dana Wolf from the Hadassah Hospital. So just to kind of explain, the medical hospital of the medical school of the Hebrew University actually shares a location with Hadassah Hospital. there's like a round door that separates between them. So it was actually very convenient to do this. And Dana Wolf has actually been amazing throughout this process. And what they've done is they build a robotic facility that will enable a much larger scale of diagnostic testing. So this robotic facility is actually there's over 50 volunteers from the university. So these are students, professors, staff members that are usually just conducting research at the Hebrew University. They are coming as volunteers, working 24/7 in shifts, and they're doing around 3,000 tests daily. So just to explain, this is around 20% of the tests that are done in Israel daily. So 20% of the tests is done largely by this amazing team of scientists, and that so far they have done more than 40,000 tests. So this is a setup we're coming into, and this is important because it means that we have access to samples, and also we have a place to test new diagnostics that we're doing. That actually on the daily basis, the person running this facility is called Roni Ben-Ami, and she's a student, a medical student herself at the Hebrew University. So she's now running this staff of 50 volunteers. And I'll soon tell you what we've been doing, but actually a lot of labs have been tuning into this amazing collaboration and developing many different tests and medications, etc. So also Roni Ben-Ami herself, along with many collaborators, has already developed a test that's been already tested and widely used in Hadassah for widespread screening of populations. For example, in the Hadassah Medical School, every week they're screening the entire staff to make sure no one is sick for COVID-19, and that's been really critical for preventing the spread of the disease within the hospital. And what they're doing is something very simple. For every sample, instead of starting with one individual, they're starting with 8 individuals. So it means that the protocol they're running requires 8 times less material and is 8 times faster. The problem is that, of course, if a person is sick, we wouldn't know which of the 8 people are actually sick. And also but they are able to go back to these 8 individuals and then test them individually. So this has been a really great trick, but the problem is it's using the same materials that's been used in the regular testing. So we wanted to do an additional improvement for that. So now I'll tell you about the technology we've developed to improve, which we call an improved homemade COVID-19 diagnostic test. And what we tackled is, as I said before, I'll just remind you, we tackled one step within this current diagnostic test. It was very important for us to keep the entire pipeline the same, so labs around Israel can very easily adopt our diagnostic test because they're only replacing one very specific step, which is the cleanup of the RNA. So as I said before, currently this is done by buying very expensive kits that's been produced by commercial companies around the world and not in Israel. So we need to import these commercial kits and then use them to extract RNA. And throughout the world, it's been a huge, huge shortage in RNA extraction kits. So what we've done is we've actually used a material we frequently use in the lab to clean up RNA or other genetic materials, which are tiny magnetic beads. So let me explain how it works. We have within our tube with our lysed RNA, we have tiny, as you can see here on the left, we have we add tiny magnetic beads. These are these brown circles that you see. And the magnetic beads have a very unique property that they actually attract the RNA molecules. So because they're magnetic, we can very easily hold these magnetic beads and just wash off all the the mess that comes with the RNA, and then use water to actually extract the RNA molecules from the beads. So actually we're skipping the need for RNA cleanup, and we're directly capturing the RNA molecules directly from this the lysis buffer. So what we've done is we've replaced a lengthy process that takes hours with a relatively short 30-minute step. And most importantly, we've made this protocol in a way that we can easily produce in every lab. We can easily produce these magnetic beads and all the reagents that are needed, the materials that are needed throughout this process. So this becomes significantly. So looking at this in this usual test, we're now integrated our RNA extraction protocol using the magnetic beads into the regular pipeline, and we made this whole pipeline significantly cheaper, faster, and very importantly also all made from homemade reagents. And when I mean homemade, it's actually lab made. And importantly, we made this protocol available also on robotics. So you could see here a little movie. I hope it works, yeah? So you hear Ayelet speaking in the background. What you see is how this robot is called a liquid handler. It can handle 96 samples at once. Right now what it does is it adds the samples to the beads. Now it's going to dispose them. You're going to see the beads going up and down in the tubes while it's mixing them. This will happen in 1 second. And this is basically the gist of the protocol. So you could see how easy it is to operate. Once the robot is set up, you can any handler can actually build it. I'm going to stop this movie and let's continue. Oh, I see the movie started again. Okay, now we're good. So I hope you can hear me. Let me just 1 second. Okay, so can you still hear me?
Yes, we can. We all hear you.
Okay. So my computer froze for a second, so I'm happy we're still back together. So I removed my video for a short while, and I'll turn it back on just to make sure. So I hope now you got a clear picture of what we're doing, and we actually tested our protocol for over 600 clinical samples. So I couldn't not include a single graph in this whole talk. So this will be my graph. Where we're showing that our method is both accurate and sensitive, and we compared it to existing methods with commercial solutions, and we show that we get the exact same predictions for every individual if he's either sick or healthy. So this has been a really great, and the feedback we're getting around the world has been absolutely amazing. But it's still not enough, at least for us, because we're still, our protocol, which is already much more efficient, is 4 to 10 times more efficient than existing protocols. So it does solve completely the problem of the missing reagents, but it does not solve other problems such as the scalability, because 10 times faster is already a huge improvement, but it's not enough if you really want to routinely do huge surveys of hundreds of thousands of people in a population. And I know for bigger countries than Israel, they will actually need to scale up much, much more. So what we are, Okay. So our next step is to try and scale up the COVID-19 diagnostic by using a different approach. So let me just explain the problem. The problem is that in the current protocol, we're limited to 96 samples at a time. So these plates that I've been showing you and the robot has been working with, which is quite standard in many countries around the world, works with 96 samples at a time. So this sounds a lot, but let's think of 100. How many plates would we need to do 1,000 samples, for example? It starts to scale up. Now let's think of 10,000 samples, which would be even like the bare minimum for diagnostic tests. You could see here I'm actually putting the amounts of plates needed for 10,000 testing. I think you could already get a sense that this is not scaling up very easily. And the problem is that we are processing every single individual in a separate tube throughout this process. So there's no way we could really scale this up to 100,000. The only way to scale this up is actually to pull people together into a single tube. And this is actually the basis of our idea, which we call a tag and pull sequencing technology, where we're actually replacing these the last steps of the PCR, we're actually replacing with a single tube, which will contain all the thousands of samples together, which will all be loaded on a high throughput sequencing machine that will give us the answer for thousands and thousands of people. So where is this idea coming from? I'll actually get back to, if you remember my first slide, I told you a little bit about my background as a neuroscientist. And I told you that what we've actually been doing is developing genomics technologies. And in fact, what we're doing here is very similar to what we've been doing in the last few years to study the brain. So in order to study the brain, which is a highly complex tissue, has so many different cell types, we needed to develop a technology that would enable us to measure RNA molecules, the same molecules we now want to measure for the virus. We wanted to measure RNA molecules from humans in every single cell in the brain. And we had to do it not for 1, 2, or 100 cells, but rather to tens of thousands of cells. So we developed a technology, and actually simultaneously around the world, several people have developed similar approaches that enables us to take 10,000 cells and measure RNA molecules in each of these cells individually. So let me just kind of give you in a nutshell how this type of technology works. You can see here at the left side, you can see cells that are encapsulated in a tiny drop. And they are encapsulated together with a small microparticle, which is very, very similar to these microbeads I just told you about that can capture RNA. And then after they're captured together, we actually release the RNA from the cells, and all the RNA are binded to the beads. And the whole the big idea here is while they're binding, they're not just binding like I showed you before, that we're binding RNA to the beads in order to capture the RNA. We're also adding something to these RNA molecules, which is a small barcode. So because every RNA molecule now has a barcode, we don't need to separate the samples anymore, but rather we can pull all the samples together into a single tube and use genomic sequencing technologies as a readout for our libraries. And what are we actually reading in the sequencing technology? We're reading the sequence of these RNA molecules, but we're also reading the tiny DNA barcode we added, which enables us to know from which individual this molecule came from. So just putting this all together, this is exactly what we're doing now. We're starting with samples gathered from individuals that are put into the slices buffer, but then instead of continuing working in 96 well plates, we're actually pulling hundreds, and in the future will be thousands of samples together in tiny tubes. And actually, between 10 to 100,000 samples can be loaded on one sequencing machine, which means that one sequencing run can already give us the answer very accurately to 100,000 individuals. We can know if they're sick or healthy. And the sequencing technology actually opens up really exciting ideas for scientists like me. We can now try to think, can we track the spreading of the virus, the sequence of the virus, and other things that the traditional qPCR machines can't give us. But not even going that far, the most important part right now is we have a way to pull hundreds of thousands of people, of samples gathered from people together, where every individual has a unique barcode that at the end, we put them all on a sequencing machine, which tells us yes or no, does this person have or doesn't have a viral RNA based on its unique barcode. So you can see here actually down at this part that we're actually integrating both of our technologies together, because our first step is to extract the RNA from the individuals, which is based on these tiny magnetic beads where we use to capture the RNA, but at the same time, we're also adding the barcode to them. So I hope that was clear. And this is really the exciting results. And just, I forgot to say where we are at this stage of the project. So we have actually already been using clinical samples from the help of Dana Wolf to show that we are able to apply this exact protocol that we developed on clinical samples and that it's working. We get positive results when the person is positive and negative results when it's negative. But so far, we've only done it on around a few hundreds of people, and we need to now go to the second phase of massive testing of thousands of people in order to implement this. So I'll just end by thanking again this amazing team of scientists that I've been so lucky to work with, Professor Nir Friedman, of course, all the Friedman lab and the Habib lab, Professor Dana Wolf and Professor Yuval Dor from the medical school, and our funding sources, which are always very critical. So we have our scientific funding sources, which we've been actually using so far to do these projects. And we also got a generous donation from the Rothschild Foundation, especially for the COVID-19 testing. And of course, Viviana Kazam for her amazing contribution to science and to my lab. So that's it, and I'll be happy to answer any of your questions.
Yes, and I will now open the microphone of Anat Adi Shapira. She raised her hand. Anat?
I think she's muted. Anat, if you hear us, you need to unmute yourself.
I unmuted her already. Yeah? Okay, so let's go to Nadine Biazini, our friend from and president from Switzerland. Nadine? Nadine, can you talk? Hello? Okay, seems that our microphones.
Can you hear me now, maybe? Yeah.
Nadine, you can speak. Hi.
Okay, good, great. Hello, Mara. Thank you so much for this really interesting and eye-opening talk. I'm wondering about these beads, actually. How do you fabricate beads which attract a specific virus, which seems to be in this case the corona 19? Can they attract other viruses, or how does this work a little bit in more detail?
Yeah, that's two actually great questions, because I think one requires a little clarification, I think, on my side. So these magnetic beads are actually something we use routinely in genomics labs because they have because of their genetic, electrical charge and the electrical charge of genetic material like RNA and DNA molecules. So we could use all kinds of specific chemistry with specific buffers and create the conditions where these magnetic beads attract any genetic material of any type. So what we're actually extracting is not only the viral material, we are extracting any RNA molecule that comes within this swab. So also like it could be RNA from epithelial cells in our mouth, for example. So human RNA, it could be also many other pathogens, bacteria and RNA. And actually that's why we're also excited about this method, that it could actually be adopted later to testing of any virus or even any pathogen as well. It's not specific to COVID-19. How we're generating the beads, that's actually, we're not exactly generating the beads themselves. We're buying them, and it's actually quite cheaply. For example, we just bought a liter of these beads, which can be used for over 1 million tests. And what we're doing is we're adjusting the beads with a very easy protocol, which we've already published, in order for them to attract the RNA and release it. So the way they're coming right now, they cannot attract the RNA molecules, but and this is all detailed in the protocol we've already published.
Okay.
Congratulations. Can I ask another question? Can it also destroy this virus in some way, or could it be used in a...
That would have been great.
Why doesn't it? Why can it?
Yeah, I also do not recommend to drink or inject anybody these beads or with any other materials. But no, unfortunately, that's not the case. So what actually destroys the virus is if you maybe recall in my slides, is the lysis step. So we're adding very strong detergents that completely ruin the membrane of the virus and opens it. And that's the part that actually destroys the virus. So it ruins proteins, it ruins the membrane, it does not harm the RNA, and that's why we can then later extract the RNA from all the other mess that we've created by the lysis buffer.
Okay. So now we have a question from Josh Kleinman. Josh?
Yeah. Josh, I do think you need to press the unmute.
Yeah, I just did.
Okay.
Thank you so much for such a clear presentation. And I'm not knowledgeable enough, like our previous questioner, to ask specific questions on the testing, but being someone who is active in the field, I'd like to ask you in a different direction, when you think, in your opinion, we will see a vaccine that will work. We hear 12 to 18 months. I've heard 2 to 3 years. In your opinion, what do you think we'll be looking at in the future?
Yeah, I mean, this is, unfortunately, I don't have an answer for that. And to be honest, I'm also not an expert on the vaccine development. I do understand very well the complications. So it seems very hopeful because there are several already clinical trials ongoing. But I think what's really important to understand that it's still, I mean, the minimum time it will take is another 6 months, and that's because of the length of clinical testing. But it is important to understand that a vaccine might not be a magical solution. And because just think, for example, the regular flu vaccine does not give us 100% protection from the virus. It just reduces the severity of the disease. So it could be that reducing the severity of the disease would be enough, but we actually don't know a lot about the COVID-19 virus, and we don't know how efficient a vaccine could even be, how long will people stay vaccinated, and that's why I think all these solutions should come at once, and we will need to keep doing these massive testing for a long time. So that's why we need a very cheap way, an efficient way of doing it. So I think all of these solutions come at once. We can't just wait at home until the vaccine arrives because it might not be enough.
Thank you.
Okay, now we have Murai Cohen. Murai, please open your microphone.
There, can you hear me now?
Yeah.
Yes.
Hello? First of all, Professor, thank you. Your approach is absolutely brilliant. And its particular brilliance is in its elegant simplicity, if I may say so. I have two questions. The first is, in your sequencing, are you able to distinguish between the different strains or substrains that we're seeing now, that the virus is so prevalent that it's already starting to show separate signatures? For instance, here in Central America, we already have 6 separate strains that we've identified, even though we haven't done a whole lot of testing yet.
Right. So I just hinted about that at the end of my talk. So I see you're a real expert, so just I'll explain a little bit. So we're already seeing changes in the sequence of the virus, of the sequence of the RNA, and this is what actually determines the different strains of the viruses. And this could potentially lead to the efficiency also of vaccines and other attributes of these viruses. So when we're doing the traditional test that's based on the real-time PCR, we're only getting an answer yes, no. What we're now suggesting to do, which is based on sequencing, we're actually reading the exact sequence of the genetic material of these RNA molecules. So potentially we're actually going to learn much more. Currently in the first version of this protocol, we're not reading the entire length of this molecule, we're reading a very short sequence, which will help us just to identify the virus. But at the end of my talk, I hinted that we could actually do so much more. And one of the first things we're already designing to do, but we don't have the protocol up and running yet, is to read significant more portions of this virus so we could do exactly what you're saying. And for every individual, we could say not only if he's sick or healthy, but also what strain of the virus he has. So it could be helpful for us to understand how quickly the virus is changing, but it also can help us to identify chains of infection. So we could kind of try to understand for every sick individual, where does this virus originated from, which could help us in also prevent the infection to others. So this is the potential of you moving from PCR to sequencing. These are one of the advantages.
My second question is, are you interested in collaborating with other labs around the world so that you can in fact see sequences from every strain that's happening simultaneously in the world?
Yeah, of course. And we really are interested in open science. It's been our policy in like a lot of other labs these days. Every protocol we develop, we immediately publish it, and we would love for other labs to implement it as quick as possible and give us feedback, and so we could actually see, compare results from around the world that would really improve our science.
Great. I'll send an email to try to start that process. Thank you so very much.
Thank you.
Okay, we have now Eric Simon. Eric, you can open your microphone.
Yes. Hello? Yes, you heard me?
Yes, we hear you.
Okay. Great work. I have a question. You have many cycles of thermocycle in your process?
We are using PCR machines in our process to generate the library. However, this is done after we pulled already at least 100 samples together. So that effectively, that means that if currently we're working with plate of 96 samples, you could potentially double that by 100 and with the same capacity of machines.
Yes, but in cycle,
machine, yes.
in temperature cycle, you have many, or you have only one cycle?
I think I don't fully understand your question. Maybe it seems very quite. You seem to also be a real expert. So maybe I would invite you to write me an email and we can discuss that forward, because I think it will be hard to drill down all these details.
Okay.
Yeah.
Thank you. And I think you can again raise your hand, but we can also go to the chat. There are a lot of questions there too. I see Rafael Goldstein. He's asking if these beads are commercially available from GE Healthcare or Cytiva. Sorry for the pronunciation.
Yeah. So these are exactly the GE beads that we're using. There's also a commercial version of these beads that usually we've been using in the lab. Right now we've bought a different version from GE, and it's all written in our protocol actually. But so you could use even you don't need to do the homemade version. You could also buy a commercial one, but the beads itself come from GE.
Okay. And now another question here is, how do you imprint the barcode on the unit?
Yeah, that's one of the challenges. It's actually not that complicated. The barcode is a DNA molecule. So we just generate a random and that's used a very known technique where we generate random sequences of DNA. And because they're random, they're also each unique and each different from each other. And we use simple biological steps in order to integrate the DNA barcode combine that with the RNA sequence. For the more biological crowd in the audience, we're basically doing a reverse transcription step followed by a PCR step in order to integrate the barcode into a cDNA molecule. So that's quite fairly standard. And the trick is to just combine this with the right barcodes and the capture of the beads.
Okay, so Dvora Feder, she's asking, did you compare the sensitivity of the qPCR assays with the different RNA cleanup methods using a standard RNA samples?
Yes, I actually had one slide, that graph I showed with that was actually the data where we compared over, I haven't showed that, but we compared by now over 600 samples, showing that we have the same sensitivity as current methods used in labs. And we're actually doing a second round of optimization now to even increase this sensitivity, because also current methods, when somebody has a very, very low amount of viruses, it's hard to detect if he's sick or healthy. We get the borderline results. So we're trying to actually also improve on that. But currently our method is similar to we compared it to 3 different extraction methods. So of course, any person who would like to apply this method, we suggest to first compare it, of course, to your in-house method.
Okay, we now have Aliet Jeman. I open your microphone. You can ask your question.
Thank you. Firstly, it was an amazing talk. and I just would like to salute that. And I have two quick questions. How long is the testing process in your lab? And second question, could it be applicable at the entrance of a plane or an airport?
Yeah, so thank you for this question, because it's a good point to clarify, actually. So when we say our method is much more efficient and faster, we're comparing it to a lab, a test done in a lab. So it's not, it's still a test that needs to be done in a lab. It needs laboratory equipment. And in the efficiency actually enables that lab, instead of processing, for example, 1,000 samples a day, can process 10,000 samples a day. So that could be a huge improvement for a diagnostic lab. But it's not the type of test that you could do by just you know, spitting it into a tube in an airport, for example. But there are. But going back to my message about scientific innovation at times of COVID-19, you could see around the world many scientists actually developing such technologies for rapid testing. So a lot of time it actually does compromise the sensitivity of the test. But I think the real solution requires both, right? Because there are situations like preventing a sick person from going on an airplane where you need a very quick test to be applied, and other situations where we want to do a survey of 100,000 people in a population where we can actually use, we don't need to give answers in 15 minutes. That's not our limitation. Our limitation is the scale. I hope that answers your question.
Yes, thank you very much.
Okay, let's go back to our chat. Are you using RT lamp method?
Yeah, so actually my colleagues from my postdoc laboratory at Frank Zhang's lab have been developing other methods, and one of them is using the RT lamp method. The problem is that it actually doesn't fit the way we currently collect samples in Israel. It requires, actually, there's no RNA extraction method. They're actually, after swabbing, they use heat inactivation. So currently it actually cannot be applied. So we're developing an alternative method to that, but it seems like also a very promising approach that I hope will be actually applied as well.
Okay, and Irene Minkowski, she's asking cost and availability of the test.
Okay, so the cost, so I actually presented two tests, right? The first was just a plug in to the current test, which reduces the cost of generating, of just the RNA cleanup. So from a few, just for that step, from a few dollars per person, it could be a few cents per person. So it's a very significant reduction in cost. But our more general idea of the RNA sequencing, you could think of that the entire process will be a few cents for every individual, because we're actually replacing all of these components within the system. But this will only be cost effective if we are indeed doing tens of thousands of people. So it's a different, completely different way of thinking. Regarding the availability, we've already published our protocol. We hopefully were planning to publish our second protocol within a week. But we're not a commercial entity. We do not provide the materials, but rather we openly provide our science for other people to implement them around the world. And we've indeed been getting approaches from around the world to do that. Hopefully, if a company would actually start producing that, it could be also a way to produce these
I think we have time for one more question from Mark Salem. Can this test identify those individuals that have in the past have had the virus but now have developed immunity?
No, so that's something that any test that's based on that is trying to identify the RNA of the virus can only identify somebody who's sick right now. And this is actually where antibody testing might come in. It's where when we're doing antibody testing, you can potentially say if somebody is currently trying to fight the virus based on the type of antibodies that he has, but you might also be able to say if he has been exposed previously to the virus and he holds the antibodies to fight it. So it is a little, the world of antibody testing is quite complex, but very promising. The problem is you probably can't identify, it's not clear yet if you can identify just based on the antibodies, somebody who's at the initial step of the stage of the viral, of the exposure to the virus. This is where the RNA tests are actually highly efficient, and it's unclear how efficient the antibody testing will be. Also for asymptomatic people that carry the virus but have no symptoms, it's not very clear how well can we detect them based on their antibodies only. Mara, you're muted.
Sorry. So I was saying thank you very much for your great lectures. I see there are a few questions about the recording, so I'm going to send the recording tomorrow to the mail that you registered with. And thank you again to all the participants. We will have another great webinar tomorrow from our faculty in Rehovot with Masha Niv. So I hope to see you there too. Thank you very much.
Thank you for this opportunity and for listening.
Thank you.
Thank you. Goodbye.